Hello and welcome to today's webinar. Our subject today is on automating IDT xGen hybridization capture of DNA libraries on the Biomek i7. Our speaker is Enrique Newman. Enrique is currently the Field Marketing Manager for Beckman Coulter Life Sciences. He studied biology at the University of Santiago de Compostela in Spain. And during his PhD at the University of Edinburgh, he focused on the molecular processes in plant cells. Since then, he has worked on several positions related to automation solution for genomics laboratories. Thank you, Enrique, for this webinar today.
Thanks a lot for the introduction. Today, I'm going to present to you the solution that we have on the automation of IDT's xGen hybridization capture of DNA libraries on our Biomek i7 Hybrid Genomics Workstation, so on our automation platform. My name is Enrique Neumann, and I'm the Market Development Manager for Genomics and Automation in Europe. Just a short disclaimer as we begin with the presentation. I want to let you know that all instruments and reagents used for these purposes and that are going to be shown in this presentation are for research use only and not for use in any clinical diagnostics. I'm going to give you a short introduction to Beckman Coulter and who we are. So we are a global company that we've been out in the market for many, many years now, and we are one of the most recognized providers for life sciences solutions. We have, as you see here in the map, where we have locations all over the world. We have a very global presence, and we have above 3,000 employees worldwide. Beckman Coulter Life Sciences has a very wide range of solutions for life sciences that go from flow cytometers to solutions in centrifugation, but today we're going to center our presentation to two of our product lines. The first one is our liquid handling solutions that range in volumes that go from 2.5 nanoliters up to the milliliter scale. So as you can see, we have a very big range of instruments here. And we start with the Echo. So the Echo is an acoustic dispenser. So it's an instrument that it's able to move from plate to plate droplets of liquid using sound waves. We have as well a whole range of our classic flexible liquid handlers that it's what we're going to be speaking the most about today, so the Biomek. And last but not least, we have as well the Biomek Ingenious, which is a compact solution that has been designed for NGS library preparation. It's super easy to use and it needs no programming skills whatsoever. Our reagent portfolio is the other thing I want to mention. So probably you know AMPure beads. You've heard about them if you've done NGS library preparation. So AMPure beads are used to clean up DNA, RNA, and as well to do size selection of DNA during the library preparation process. So Beckman is a company that produces these beads. Our reagent portfolio, it's all powered by the solid phase reverse immobilization or SPRI technology. And of course, the product that is known the best from this are our AMPure beads. So SPRI, this technology has the ability to provide very reproducible size selection results for NGS applications, and they are very flexible to in a way that we have a very wide range of nucleic acid extraction kits that go from DNA isolation to RNA isolation and total nucleic acids from a wide amount of samples like cells, tissue, blood, and even FFP samples. If you want to know more about these, I'm more than happy to answer any question you might have. And to tell you a bit how we work together with our customers, in order to optimize their extraction needs. Let's move to the central matter of this presentation. Actually, here I'm going to start presenting the solution that we have on our automation platform for automating IDT xGen hybridization capture of DNA libraries. So the whole process can be automated with our platforms from the very beginning of the protocol until the very end. And what are the advantages of being able to automate your library preparation process? Well, it increases efficiency, it provides flexibility, it reduces hands-on time, and it increases the throughput you might have. And as well, all of this is thanks to the quick install with our ready-to-use methods. Something that I want to highlight here that I think it's very important. It's one of the main assets of automating your library preparation process. It's how you improve the reproducibility of your results. So think that library preparation is a very complex process. It's something that it takes time. It's a lot of manual work. And actually the results are very variable depending on which hand is holding the pipette during the library preparation, and even how that hand is having the day. So actually, with the automation of this process, we have the great advantage that the instrument behaves every single run in the same way. So samples will be treated exactly the same way from batch to batch, increasing our batch to batch and sample to sample reproducibility. And of course, as well, reducing the risk of errors, especially the cross-contamination ones. Now let's have a look to the instrument that is building those libraries for us. So this is the Biomek i7 Hybrid Genomics Workstation. So I'm going to explain you and walk you through bit by bit how this instrument is built and what can it do. So this instrument, the Biomek i7 Hybrid Genomics Workstation, actually has two pipetting arms. The first one, it's a multi-channel head. So this multi-channel head, it can be either a 300 microliter or 1200 microliter multi-channel head with a range that goes from 1 microliter to that maximum that I shown, and it's going to work either with 96 or 384 channels working in parallel and processing the whole plate at once. The second pipetting arm, it's what we call the span-8. So the span-8, it's actually 8 independent channels that are independent in position, in volume, and as well they can separate. So this really allows us to have the maximum flexibility, and we can do normalization, we can do cherry picking, we can move samples or reagents from tubes to plates, from plates to tubes without any problem. And this works with either fixed or disposable tips. In the case of NGS library preparation, we are using our disposable tip option to make sure that we are minimizing the risk of cross-contamination during the process. The box that it's the instrument, it's safe. It's a very spacious open platform that it's designed actually in a way that allow us to integrate other elements, other devices that we might want to have, either on the deck or off the deck. So it can be that we have them on the sides and, for example, have a thermocycler, a plate reader. We can have a full range of elements to make our solution easier and more walk away. We can move elements from one place to the other on our deck. Thanks to our 360 rotating gripper that has offset fingers that gives us access to virtually any position on the deck. So we can, for example, move plates in and out of a magnet, or we can move them in and out of a thermal cycler, or to an incubating position, or to a shaker. So This is thanks to these grippers that are attached to each of the pipetting arms that the instrument has. The deck as well has a very high capacity with up to 45 positions for our tips, our plates, and whatever that we will be needing for this library preparation process to make sure that our protocols are as walkaway as possible. In the Hybrid Genomics Workstation as well, we do have orbital shakers and peltiers for incubations to make sure in one hand that our reagents can be kept at 4 degrees so they are cold and safe throughout the whole process, and as well that we can do all the incubations that are necessary during the protocol on deck. So how does this automated method looks like? Well, here we have a lot of numbers, we have a lot of data, but what I want you to see is that actually in the last row, we have the total time for this hybridization protocol. And as you see, the time that it's necessary for processing different sample numbers, so 24, 48, or 96 in these cases, remains practically unchanged, is very similar, which really shows how we can improve our throughput in the lab thanks to automation. So as you see as well in the first row, we see the instrument setup time. We have a very low setup time. It's very easy to set up the instrument and get it ready to run. And actually, we can process really a very big number of samples without major efforts from our part. We've seen how the instrument looks like, but I want to show you as well how the software looks like. So in my opinion, one of the biggest assets that we have on the Biomek, it's actually the software. It's very user friendly. It's very easy to understand how to use it. And with these preset methods that we have something that is called the Biomek method launcher. So the Biomek method launcher makes it very, very easy for the user as we select the method that we want to run, as we have here in the first image on the left. And then in the central image, what we see, it's actually the different options that we have to run the method. So we are actually setting variables like the number of PCR cycles that we want, or how we want to carry out the pre-hybridization of our samples. Finally, this Biomek method launcher is going to give us a guided labware setup. So it's going to guide us through on how to set our work table, where we need to put each of the elements, where we need to put the tips, the plates, the different reagents, and it's even going to tell us which volumes are necessary for the sample number that we are processing. So as you can see, it's very, very easy to set up and use the instrument and get ready to run. And last but not least, I want to show you a few sequencing metrics of samples processed with the Beckman i7 with this IDT method. And actually, the take-home message, what I want you to remember is that we have in all the graphs, we have different metrics like the duplicate percentage or the target coverage. And we have in each of the graphs on the left historical data of samples processed by hand. And on the right side, we have samples processed with the Beckman i7 Hybrid Genomics Workstation. And what I want you to notice is that that cloud of dots that we have, in the case of the samples process with the automated method, it's a lot more tight. So that means that we have a lot more reproducibility from sample to sample, as I told you before, because every single time the samples are processed in exactly the same way by the instrument, avoiding all the errors that can be generated when we are working by hand. So we've reached to the end of this presentation. I'm more than happy to answer any questions you might have.
Thank you, Enrique, for this presentation. That was really a nice overview. So we do have some questions. The first one is, are there any other kits from IDT that are automated?
Yes, actually, so both the library preparation and the hybridization kit are automated, and we have ready-to-use scripts. But I have to highlight that any other kit that you might want to automate, we have a very strong applications team that can help you put that into your robot. So, for example, if you're interested in using the methylation kit or any other IDT kit, we are more than happy to help you get there.
Thank you. Another question is, can you run other protocols on the same robot, like the Biomek i7?
Yes, actually, one of the beauties of this instrument, of the Biomek i7, is that it's completely customizable. So it can be adapted to run whatever you need. So may that be another NGS protocol that needs pretty much the same layout, or a complementary type of application, for example, quantitation and normalization, or pooling, or actually whatever you can think of. The sky's the limit here. So yes, the instruments are completely customizable, and we can implement whatever protocol you might want to run.
Thank you. And then a last question to finish this up is, could this workflow that you showed us, could it be adapted for other sequencing platform?
Yes, so I know that with some tweaking, it's possible to use it for other sequencing platforms that are not necessarily Illumina. So the answer is yes, it's possible.
Thank you very much, Enrique.